A tree turning from green to gold to crimson is a visible outcome of precise pigment breakdown and synthesis within each leaf. Leaves are green in summer because chlorophyll absorbs red and blue light while reflecting green. During autumn, daylight shortens and temperatures drop, signaling trees to prepare for winter. The first step is the breakdown of chlorophyll into colorless compounds, removing the dominant green pigment. Without chlorophyll, the underlying carotenoids yellow and orange pigments become visible. Simultaneously, anthocyanins are synthesized in some species, adding red or purple hues to the leaf surface. The synthesis of anthocyanins is driven by light exposure and sugar accumulation within the leaf cells. These pigments are stored in vacuoles. Their concentration changes as the chlorophyll's pH shifts during senescence. As the leaf ages, cell walls thicken and water content decreases, concentrating pigment molecules. The combined effect of exposed carotenoids and newly formed anthocyanins creates the vibrant autumn palette. Eventually, all pigments are degraded or transported away, and the leaf falls to the ground. Pause 0.8. Chlorophyll molecules inside chloroplasts absorb primarily blue and red wavelengths, while reflecting green light. During photosynthesis, absorbed energy drives the conversion of carbon dioxide and water into sugars. The pigment structure contains a porphyrin ring that binds magnesium at its center. When daylight wanes, the plant reduces chlorophyll production and slows its breakdown. As chlorophyll levels drop, previously hidden pigments such as carotenoids become visible. Carotenoids absorb blue light but reflect yellow, orange, and red, giving leaves a warm hue. In some species, anthocyanins are synthesized later in the season to add deeper reds. The combined effect of pigment loss and new pigment synthesis creates the familiar autumn palette. Microscopic images show chloroplasts shrinking and vacuoles expanding as pigments redistribute. Thus, the disappearance of green is not a loss but a reveal of other light absorbing molecules. Scientists measure chlorophyll decline by spectrophotometry, quantifying absorbance changes across the visible spectrum. Pause 0.8. In late summer, the plant's chlorophyll production slows as daylight hours shorten significantly. As chlorophyll degrades, it releases its green pigment into the leaf matrix. The remaining carotenoids beta-carotene and lutein were already present, but masked by green. These yellow-orange pigments are stored in plastoglobuli within chloroplasts. When chlorophyll fades, the carotenoid structures become visible to our eyes. Beta-carotene absorbs blue light and reflects orange, while lutein reflects yellow. The combined reflection produces the bright hues that signal autumn's arrival. This pigment breakdown is a controlled process regulated by plant hormones like abscisic acid. The visible color change also signals to insects and animals that the leaf's nutrients are diminishing. Thus, the hidden palette of carotenoids turns the forest into a natural art gallery. The timing of pigment exposure is synchronized with the plant's internal circadian clock. As days grow shorter, the plant conserves energy by shutting down photosynthetic machinery. Pause 0.8. In autumn, trees begin to produce anthocyanins, bright pigments that give leaves reds and purples. The synthesis starts when light signals the phenylpropanoid pathway to activate key enzymes. Chalcone synthase catalyzes the first committed step, converting p-calvaryl-CoA into chalcone. Chalcone isomerase then rearranges chalcone into naringenin, a flavanone core structure. Subsequent hydroxylation by flavonoid 3-hydroxylase introduces additional oxygen atoms for color diversity. Dihydroflavonol reductase reduces dihydrochemopherol to leucanthocyanidin, the immediate anthocyanin precursor. UDP glucose flavonoid 3-ogalytosyltransferase attaches glucose units, stabilizing the pigment for storage. Accumulated sugars in leaf cells raise osmotic pressure, encouraging anthocyanin accumulation. Light intensity and duration modulate enzyme expression, while lower pH shifts the pigment toward red hues. Together, these biochemical changes create a visible spectrum that signals seasonal transition. The final pigment is stored in vacuoles, where its stability depends on the surrounding pH and metal ions. When temperatures drop, chlorophyll degrades faster than anthocyanins, allowing the red hues to dominate the canopy. Pause 0.8. In autumn, trees sense a shortening of daylight hours, which signals the end of the growing season. Cooler night temperatures slow down cellular processes that maintain chlorophyll in leaf cells. As chlorophyll degrades, green pigment fades and the underlying yellow and orange pigments become visible. Simultaneously, reduced water availability limits the transport of nutrients needed for pigment synthesis. The plant reallocates resources from growth to survival, prioritizing protective compounds over photosynthetic ones. Carotenoids, which are always present in leaves, shine through once chlorophyll is gone. Anthocyanins may also be produced in response to light and temperature cues, adding red hues to the palette. The combined effect of these pigment changes creates the characteristic fall foliage display. A graph overlaying temperature, photoperiod, and soil moisture against the leaf color intensity illustrates how each factor contributes. Understanding this mechanism helps explain why some regions have more vibrant autumn colors than others. Pause 0.8. When daylight wanes, leaf cells begin to shut down photosynthesis and start breaking down chlorophyll. The first enzymatic step removes the central magnesium ion from chlorophyll, forming phyophytin. This reaction is catalyzed by the enzyme phyophytinase, which cleaves the metal binding site with remarkable specificity. Phyophytin then undergoes a series of oxidative reactions that open the chlorophyll ring and release the phytol tail. Chlorophyllide deoxygenase CaO introduces an oxygen atom into the porphyrin ring, converting it to phosphoride. Phosphoride is further degraded by hydrolases that break the remaining bonds, yielding colorless catabolites such as chlorophyllide. These catabolites are transported out of the chloroplast into the vacuole for storage or eventual recycling. Because the final products lack the conjugated double bond system, they no longer absorb visible light and appear colorless. The disappearance of green allows underlying pigment carotenoids and anthocyanin to dominate the leaf trade's visual palette. Thus, the enzymatic breakdown of chlorophyll is a controlled, stepwise process that directly drives autumnal color change. Pause 0.8. During autumn, trees initiate senescence by shutting down photosynthetic machinery in their leaves. Chlorophyll molecules are first degraded, releasing bound nitrogen and phosphorus into the leaf cell. The freed nutrients then travel through specialized transport proteins toward vascular tissues for redistribution. Simultaneously, carotenoids and anthocyanins become more visible as chlorophyll fades, signaling the end of energy capture. The plant uses this visual cue to coordinate leaf abscission, ensuring minimal loss of valuable resources. Once the leaf detaches, remaining nutrients are absorbed by root systems through a reverse flow of xylem sap. This nutrient resorption reduces the need for new mineral uptake from soil during winter dormancy. Energy that would otherwise be spent on synthesizing fresh leaves is redirected toward maintaining root growth and storage. Thus, pigment breakdown not only creates autumnal color but also initiates a critical recycling loop. By reallocating nitrogen and phosphorus back into the tree system, plants prepare for spring growth with fewer external inputs. Pause 0.8. Leaves change color when chlorophyll degrades into phyophytin and then catabolites, revealing carotenoids that remain yellow-orange. In response to short photoperiods, cooler nights, and reduced water availability, trees synthesize anthocyanin pigments that can appear red or purple through light dependent pathways involving sugar accumulation and acidic vacuolar pH. These pigment shifts are part of a broader senescence program, where nutrients such as nitrogen and phosphorus are reabsorbed from the leaf into the tree trade's root system before abscission.